Assalamualaikum warahmatullahi wabarakatuh. Hi everyone, welcome back in the biology class with me, Miss Mai. So today we are going to learning about the photosynthesis. As we already learned in the LMS and also in the theory that the plant is make their own food with the photosynthesis. Let's see the formula of the photosynthesis. Okay, we already learned that photosynthesis is need the source of photosynthesis. The source of photosynthesis is carbon dioxide with a water, and also it can be helped by light and also using the pigment in the plant that's called chlorophyll. And it will be produced glucose and also an oxygen. So from this formula we can see that the carbon dioxide it will be performing or producing the glucose, and the water is a source of photosynthesis that will make the oxygen. For this photosynthesis, how we can prove that photosynthesis that happen in the plant is we'll use the carbon dioxide to produce glucose, and also we'll use the water to produce oxygen. By what? By the experiment. There is two types of the experiment that can be proved the source and also the product of the photosynthesis. One of them it's called, there is two experiment. The first one is called Salk experiment. And then the second one is called Ingenhouse experiment. So today we are going to try to make a Salk experiment that have a function to prove that the photosynthesis is producing glucose. Okay, let's start with the experiment. Okay, for proof that the photosynthesis is produce a glucose, so we can make the Salk experiment in here. So the tools and materials that we need for making the Salk experiment is we need some of plant. Yeah, the plants we can choose the plant that have like a thin leaves, because the thin leaf is we can see clearer the glucose that produced by photosynthesis in the thin leaves than in the thick leaves like this. Okay, and then we need also the ethanols and also water, and also we need a Lugol to testing the glucose. And how we can make the Salk experiment? So let's start to make the experiment in the plant itself first. Okay, so we can take one of the leaves in here in the plant, and then cover the leaves with the black carbon papers like this one, and then let it for 48 hours. Yeah, not cut the leaf first. Yeah, so let it in the plant, and then cover the plant with the black carbon papers like this, and then there is also the leaves that not covered by carbon paper like this one. After 48 hours, cut the leaves like this, and then open the carbon papers, and then after that take the leaves that already covered with the carbon papers and then boil it, and also the leaves that's not covered for become the controls, and then let it make a soft until it's boiled. Okay, and then I have the leaves that already boiled like this one. It's become softer, and then after it becomes softer, take the leaves here into a beaker glass like this one, and then add the alcohol. We use the alcohol 69% in here, and then put around like 40 milliliters of alcohol until all of the leaf is soaked by alcohol like this. After that, boil in the water like this one, but it's not directly boiled, yeah, but it's just like water bath like this one. Let it in here, and you can use the tongue, yeah, and then for make it's not really hot, and then let them until the leaves is become softened, and also they will be like, looks like a white, yeah. Why the leaf is will be like become a white? It will be turned like this one. The alcohol will be turned become like a green colors, and then the leaves is become white colors. Look at that. This is a white colors. We can compare with the previous leaves like this one. The previous leaf is have like a green color, but after we soak it into the alcohol, it's become lighter. It looks like a white. It's because the chlorophyll inside of the leaves is released by alcohol. So it's released into the alcohol, so the alcohol will be turned become a green, and then it's come out from the leaves. Why we should release the chlorophyll first is because we can see clearer the glucose that produced by the photosynthesis process. Okay? After we have this one, so we can put the indicator of glucose. Yeah, the indicator of glucose is a lugol. Okay, so in here I have the leaves that's already covered with the carbon papers, and also the leaves that's not covered in the carbon paper. When I put the lugol here, into the leaves, I'll put the lugol in the leaves that covered with the carbon papers, and then the leaves that not covered with the carbon paper. We can wait it until 5 minutes for see the result. We can directly see that the leaves that's covered by a carbon paper not indicated the glucose or the starch that consists inside of the leaves, because there is no photosynthesis that occurs in the leaves because it's covered by a carbon papers. But if the leaves that's not covered with the carbon paper, it show the blue-black color that indicated there is a starch. It means its leaves is make a photosynthesis. So if it's not covered with the carbon paper, the leaves can receive the light from the sunlight, and then it will make the process of photosynthesis happen inside of the leaves. And then when we test with the reagent of of the starch, which is a lugol, it will become a blue-black color. So the color will be changed. So from this experiment, we can conclude that photosynthesis is made or produce the glucose that's stored in the leaves by the starch, or make a starch like that. Okay, this is about the sock experiment. So for the next experiment, there is an in-house experiment, but we can go to the other videos, and then I think enough for today. So see you next time in the next video. Thank you. Bye. Assalamualaikum warahmatullahi wabarakatuh.